Welcome back, ladies and gentlemen. So today we are going to continue talking about the history of what led to scientists actually understanding that DNA, in fact, was this genetic material, the set of instructions for how our cells look and function. And up to this point, we have given you some kind of basic understanding of things that were sort of starting to lead to this idea of it's DNA or protein. And that was kind of the argument. And when we get to 1952, an experiment conducted by Hershey and Chase are going to finally solve this giant question. And remember, prior to this, most scientists are sort of on the side of those proteins are in fact genetic material because we've got 20 amino acids and a huge set of building blocks as a result, compared to just those 4 different nucleotides that we see in DNA. So Hershey and Chase are going to design a relatively simplistic experiment, and they're going to work with these things called bacteriophages, and that's what these guys are right here. So bacteriophages are viruses. They are viruses that specifically infect bacteria. And by the time we got to 1952, we were relatively good at understanding how to work with these bacteria viruses, how to work with bacteria themselves. And so it was really something that we were pretty confident in understanding. The other cool thing about these bacteria viruses is that they only contain DNA and protein. That's it. That's all that they're made up of. So we didn't completely understand exactly how these guys were working, in the sense of what material was being left in the cell and what was being left outside of the cell. But we knew that DNA and protein existed in these guys and that some set of instructions was being inputted into bacterial cells in order to make more viruses. So let me give you some background on how viruses actually infect a host cell. All right. And that's going to give you some context for how this Hershey and Chase experiment actually did what it did. So viruses are what we call obligate intracellular parasites. Okay, big crazy terms just to mean that they are parasites, so they need a host, and they are intracellular parasites, meaning they have to be within their host cell in order to replicate and get the materials that they need. And they're obligated to be within that host cell in order to do their life cycle. So each virus has a specific host range. Most viruses aren't going to be able to infect a huge number of different types of host cells. So when we look at viruses and we talk about their host range, most viruses will only cause disease in a small subset of organisms. So not every virus that infects you as a human is necessarily going to infect your dog, or your cat, and vice versa. Also, when we look within your body, those viruses often are not going to necessarily go infecting every type of cell within your body. So when we look at something like the flu, that tends to go after cells in your respiratory tract. When we look at other viruses, they may go after things in your digestive tract, right? So different viruses have different host ranges. The one that Hershey and Chase were looking at were bacteriophages that specifically went after bacterial cells but didn't infect humans, which also made them a really nice thing to study because they didn't have to be quite so concerned about their own health and safety in a lab. The other thing is that there are general features of every life cycle, or I should say reproductive cycle of a virus. They all have to get their viral genome, aka their DNA, into their host cell, whatever that is. That cell then has to be taken over to start manufacturing viral proteins, and then ultimately that cell gets destroyed as those new viruses get put together and ultimately get released. So basically, the virus goes in, hijacks that cell and all of its materials and its processes, and basically converts it into a viral factory. Once that cell is so chock full of viruses that have been built, it causes the cell to break apart, and those new viruses get released and then start going to neighboring cells and infecting them. Now, That's general features. If I look at very specific viruses, there are some very niche features, niche components, specific components to the way individual viruses work. But generally speaking, those three features are pretty commonplace from every virus we've ever studied. So when we go back to those bacteriophages, they infect their host cells, their bacterial cells, in one of two cycles, or one of two patterns, this lytic cycle versus this lysogenic cycle. So when we look at viruses that specifically are lytic viruses, all right, so these are phages or bacteriophages that culminate ultimately in the death of their host cell. And this is how we now understand it works. So that virus attaches to its host, it injects its genetic material into its host cell. Part of that genetic material that that virus puts into its host also sets up some enzyme production that breaks down the host bacterial cell's chromosome. So we chop up the host chromosome so that the host can't continue to replicate, can't continue to make its own proteins, can't continue to express its own genetics. And instead, that virus then takes over that host cell and starts using its raw materials as well as things like ribosomes that we'll talk a little bit about this unit to build proteins, to build more genetic material, will allow those viruses to get put together. And then once that cell is so full of its completed viruses, it will actually cause the cell to lyse or to break open. And when that happens, all of those new viruses that were produced are now being released out into the environment to go infect other neighboring cells. So it's very simple. Attach, insert, take over, build, release. That's your lytic cycle. Now, cool thing is bacteria cells don't have their own immune system. And why do I say that's cool? Because they still evolved a mechanism to try and defend against these viral infections. So when we look at our body as a human, we have that immune system. We have white blood cells and all these different proteins that our cells produce and tons of mechanisms to try and prevent us from getting infected by viruses or bacteria for that matter. These guys don't have that. So instead, they actually have these things called restriction enzymes. And we're going to talk about these when we get to biotechnology later this year. Because these bacterial cells contain these restriction enzymes, these enzymes recognize and cut up DNA at specific locations, and those specific locations are often connected to the types of bacteriophages that frequently infect them. So it's a great example of coevolution. Bacteria have evolved these restriction enzymes to prevent these bacteriophages from infecting them, right? So when that virus infects that cell and injects that genetic material, those restriction enzymes that the bacterial cell has evolved to contain cut up the viral DNA instead of allowing the virus to do the same to its chromosome. And if the viral DNA is in pieces, then it can't go build itself. We have taken over and utilized those restriction enzymes for genetic engineering. And so we'll talk a little bit more about those guys later this year. But essentially, for now, it's the bacteria cell's way of having a very rudimentary immune system, which is pretty neat. Okay, so I mentioned there's two different cycles. So lytic cycle is really just attach, destroy, genetic material, build, bust open. All right, so it's one virus infecting one cell and then producing a handful of cells that destroy that single bacterial cell. The lysogenic cycle is actually a little bit more sneaky in the way that it works. So that bacteriophage is still going to attach to its host cell. It's still going to inject its genetic material. But rather than just chopping up the bacterial cell's chromosome and start making new viruses, that bacterial cell actually gets forced to incorporate this thing called a prophage, and that prophage is viral DNA. So that viral DNA gets injected and then actually gets incorporated into that bacterial cell's genome. And then it just kind of hangs out there and it lets that bacterial cell go ahead and replicate, go through binary fission. And when it goes through binary fission, not only does it replicate its own genetic material, but it replicates that viral DNA right along with it. So if it goes through a single round of division, I went from one infected cell to now two infected cells. If it happens again, I've now gone from a single infected cell to four infected cells. And this exponentially grows until something in that bacteria's environment forces that genetic material to come back out of that bacterial chromosome and actually end up inducing or popping over into that lytic cycle where it starts building more viruses. So this lysogenic cycle is considerably more efficient, if you will. So when I look at my lytic cycle, right, it was a single virus infecting a single cell and out came a bunch of viruses. When I look at my lysogenic cycle, if we start there first, that bacteriophage injects its genetic material. We now incorporate that genetic material into that host. That host cell is allowed to replicate for who knows how many generations until something tricks it back over. So I may have a single virus infect a single cell, but now ends up having gone through binary fission 5, 10, 15, 20 times, and all of those daughter cells now also contain that genetic information that is viral. And so all of those cells that get produced over the, say, 20 generations of bacteria now all end up in this lysogenic, or this lytic cycle, excuse me, at the same time. And so it was one virus to one cell if all that virus could do was the lytic cycle. But viruses that can do lysogenic, it's one virus to maybe 10, 20, 30, 100, 1,000 cells that are all being capable of producing viruses once they all go pop over to that lytic cycle at the same time. So how does the Hershey and Chase experiment take this into account? So they're going to work with these bacteriophages, right? And remember I said it's protein and DNA and that's it. They're going to allow these bacteriophages to basically do what they do. But before they allow that infection pathway to occur, they're going to label the materials that that virus is made out of. So the first set of experiments they set up, they're going to radioactively label the protein. Okay, and remember at the time Hershey and Chase are doing this, they know that some sort of material makes it into that cell to give the cell instructions to make more viruses. They just don't know what that set of instructions is. They don't know if it's the protein or if it's the DNA. And that's what this experiment is going to end up differentiating. So the first experiment, they're going to label those proteins with radioactive sulfur. Okay, so that those proteins will now send off that radioactive signal. Once they've got all of these radioactively labeled proteins on these viruses, they're going to put those viruses into a petri dish with a bunch of bacteria and allow those bacteria to get infected. Now we know that those bacteriophages inject their genetic material and those protein coats kind of float off into space, float out into the environment. Once that infection was completed, they blended and centrifuged the bacteria. And what that did was basically chop up all these components. And when you run it through a centrifuge, if you've ever been on one of those roundup rides at a carnival, right, it pushes you back against the back of the machine, the back of the ride. That's kind of what happens to these test tubes, and so it forces all of the heavy stuff to fall to the bottom of the tube. Well, all of that heavy stuff is going to be all of the cellular components, right? So they looked at the test tubes and looked at the bottom, what we call the pellet, versus the top liquidy material, which is the lighter stuff called the supernatant. And they wanted to see where that radioactive material fell. Well, when they did this first experiment, and they looked at the bottom of the test tube, there was no radioactive material. It was all up at top with all the lighter material. And so this should have been kind of the end all. There was no protein that those viruses left behind in the bacterial cells. So it couldn't be the genetic material. It couldn't be the set of instructions for how to make more viruses. That being said, Hershey and Chase took it one step further just to get confirmation that they understood how this worked. So they labeled the proteins here in experiment 1 with radioactive sulfur. We go to experiment 2, they're now going to label the DNA with radioactive phosphorus. And they let the same experiment happen again. They allow those now tagged bacterial phages, right, that have the DNA that has the radioactive phosphorus attached to it. They allow them to infect the cells. They then blend the cells up again, centrifuge them again, right? Remember, all the heavy bacterial stuff falls to the bottom of the tube, all the lighter stuff stays at the top. And this time, when they looked at those tubes after the centrifugation, they saw that all of the radioactive material was down in that pellet, down in that heavy stuff at the bottom of the test tube. This was now the final confirmation that yes, the genetic material to make more viruses was in fact DNA, and that corresponded then to us understanding that genetic material was DNA. DNA was the set of instructions for how to build everything life related. How did this be capable of working? Well, if you go back to remembering your basic biochemistry, right? Why could they use sulfur versus phosphorus? Well, if you look at your amino acid structure, right? I couldn't use nitrogen, that exists in both. I couldn't use radioactive hydrogen, it exists in both. Couldn't use radioactive carbon, it exists in both. Couldn't use radioactive oxygen, it exists in both. But if I look at my nucleotide, right, my DNA, there's that phosphorus. There's no phosphorus over here in an amino acid. Now, for those of you sitting here scratching your head, well, where's the sulfur in the amino acid? The sulfur can exist in these side groups, but there's no sulfur over here in that nucleotide, and this is why this works. So if we look at the overall picture of this experiment, right? So they were able to use the radioactive sulfur because of that sulfur containing groups in the side chains or the R groups of amino acids, but it doesn't exist in DNA. So we radioactively tagged those protein coats, ran them through an infection pathway, blended them up, ran them through a centrifuge. Here's your pellet. This is all the heavy stuff at the bottom. That's all the bacterial material. And then in this case, they saw all the radioactivity up in the liquid in the supernatant, where there wouldn't be any heavy bacteria components. So it's not protein. They ran the same experiment again, this time using that radioactive phosphorus instead, right? Phosphorus only exists in the phosphate groups of the DNA. There's no phosphorus in protein, so it just sticks to the DNA. Allow that infection pathway to work again, blend it up, run it through a centrifuge, and this time, this is where all of that radioactive material was, down at the bottom with all of that heavy bacterial material, nothing in the supernatant, the liquid stuff where the rest of that protein coat would have ended up. And so we now had, by 1952, conclusive evidence that DNA was, in fact, the set of instructions for how cells are capable of doing what they do.